Hello and welcome. Happy you could join us today. This video is meant as just a brief introduction to the synapse and how neurons talk to each other. We've been talking a lot about neurons and their anatomy. Now let's see if we can take this anatomy and convert it to some sort of physiological response. The image that I've got here shows a couple of different things. First of all, it shows some synapse types. I think often we think, this is an axon onto a dendrite, and while that's very, very common, we can see here with synapse types, the axodendritic synapse is not the only type of synapse. We've also got axosomatic, where the axon actually synapses right onto the soma. And we can talk about reasons for why this might happen. We also have axoaxonic, where an axon synapses onto another axon that could play an important role in how it works. And then, of course, when we think about response types, down here, we can break this down into three different types, right? It could be an excitatory response. It could be an inhibitory response. Or it could be a modulatory response, where we're just trying to fine-tune the system. And in that case, we might not be using a ligand-gated channel like we see above, where sodium's coming into the cell, but we might be using a G-protein coupled receptor. Regardless, this just gives a kind of a brief outline of various types of synapses, and we want to make sure that we understand the basics of how this works. So to do that, let's go with the common axodendritic synapse. What I've got circled here as example number 1, right here. And let's zoom in and see what this might look like. Okay, as we zoomed in and we've looked on this, there's just a couple of basic things that we want to introduce. This is the synapse, and we can break this down into the presynaptic terminus, or terminal, that works too. Of course, we got the postsynaptic side over here. And this tells us that the direction of communication is happening from left to right. Now, one of the things, the whole basis of this, and I want to focus on making sure we understand this, this is a chemical synapse. We do have electrical synapses in the body, in the heart. That works through a different mechanism. This is what we call a chemical synapse. So we've got the presynaptic terminal, we've got the postsynaptic terminal. We then have this gap, which we call the synaptic cleft. And our whole goal for these two cells to communicate is to release from these exocytic vesicles, what we've drawn here in red, the neurotransmitter that these exocytic vesicles contain. We can, of course, see the axon of this neuron, and we also show some myelin, which is essentially the oligodendrocyte wrapping itself around these neurons. The myelin provides saltatory conduction or jumping conduction by insulating domains along the axon and allowing the speed of the axon, or of the action potential, to propagate much faster as we jump from node to node. This was reviewed in my previous video on the introduction to the nervous system. And in between these myelin sheaths, these domains, where the nodes are, this is where I have my concentrated voltage-gated sodium channels, and that's how we're propagating this action potential. Our job now is to get the cell to exocytose these exocytic vesicles containing neurotransmitter. We're not talking about what stimulated this neuron to begin with. We're just following the action potential down the axon. And now, what do we need to do? As this action potential propagates to this end, to the presynaptic terminus, we see a little bit of a twist. Whereas before we were opening up voltage-gated sodium channels, here we open up voltage-gated calcium channels. So here's my voltage-gated calcium channel. And when that opens up, of course, calcium is going to rush into the cell at these axon terminals. Calcium does what calcium does. Calcium drives exocytosis, and that's something important to remember. Now, it does this through a mechanism involving what are called snare proteins. If we were to zoom in Of course, molecularly, this isn't what they look like, but just to give you an idea, right? You can imagine a corkscrew with some arms on it. And calcium comes in and binds. And of course, calcium, because it's a divalent cation, it's got these positive charges, this causes the snare protein to essentially change shape. As like charges repel and as opposite charges attract, this forces this apparatus, this snare protein, to pull the vesicle into the membrane. It's essentially squeezing these two membranes together. And that makes sense to an extent, because the plasma membrane is a lipid bilayer. These exocytic vesicles have popped off the Golgi. They're lipid bilayers. And so we can fuse these two things. And with that fusion, what do we see? We see an emptying of the contents out into this cleft. So let's draw that down here, where this exocytic vesicle has fused with the outer membrane, and what we see is the release of neurotransmitter. So again, the action potential propagates down the length of the axon. At the end of it, what we do is we open up voltage-gated calcium channels. As calcium comes in, calcium binds to snare proteins and causes the exocytosis of vesicles containing neurotransmitter, leaking this neurotransmitter or releasing it out into the synaptic cleft. So let's zoom back out and see what effect this is going to have. And of course, the effect is totally dependent on the neurotransmitter being released. Now this neurotransmitter, if we go back and we review what we talked about before, this could be excitatory, it could be inhibitory, or it could be modulatory. Let's look at a scenario where it's excitatory. So what we've got here, we have a ligand-gated cation channel. Typically sodium. It's not always sodium. It might be specific for sodium, but maybe calcium could come in, maybe some magnesium. Again, it depends. There are multiple types. The point is, the ligand is the key to open this thing up. And of course, the ligand is the neurotransmitter. So as neurotransmitter comes and binds, what happens? We're going to see this door swing open. And as that door pops open, what are we letting in? We're letting in cations. Let's draw sodium, for example. Of course, sodium, we know, high on the outside, low on the inside, so it's got a very strong electrochemical gradient. It wants to go in the cell, and now that that door is open, we let it come in. Of course, when we start adding positives locally to the inside of the cell, that area is going to depolarize. And if we depolarize enough to reach threshold, that could stimulate another action potential in this cell. Of course, it might be that the ligand channel that we choose to open isn't for cations, but rather for anions. So here we show a ligand binding to a receptor, but this receptor, instead of sodium or cations, it's going to let in chloride. Of course, as chloride comes into the cell, we start to fill the cell with negative ions, anions, and that will have an opposite effect. Instead of depolarizing, now I'm going to hyperpolarize. Typically, not always, but typically, neurons only release one type of neurotransmitter. If that neurotransmitter, for example, is a serotonergic transmitter or neuron, it's going to release serotonin, which tends to open up sodium channels. But if I've got GABA or glycine, these tend to be the neurotransmitters that will open up my chloride channels. The question is, what do I do now with these charges? Is it possible that I can get multiple signals at the same time, right? What if I've got positives coming in on one end of the cell and negatives coming in on the other? How do I figure all that stuff out? And the cell's actually really good at doing this in a process that we call summation. To better describe summation, let's zoom out and look at multiple dendrites from the same cell. All right, here's a typical multipolar neuron. It's got lots of inputs. You can see all these dendrites and then one long axon. Sometimes there could be millions of dendrites. If we're thinking about spiny neurons associated with dopamine signaling, these things are completely arborized. Lots of places where we could have input. Let's get back to our question. What if down here at the bottom we've got a signal coming in And with this synapse, we are delivering what's called an EPSP, an excitatory, right? Post-synaptic potential. That's what EPSP stands for. And by definition, what does that mean? That means that whatever the neurotransmitter is, we are opening up cation channels. And so we have positives going into the cell. The measuring stick for all of this stuff is a location in the neuron called the axon hillock. The hillock is essentially this part, right where we start the axon. Why is this the measuring stick? Because this is where my first voltage-gated sodium channels are located. And if we can reach threshold right there, then these voltage-gated sodium channels will fire, and that will initiate the action potential and propagate it, and that's kind of an irreversible thing. There's not a whole lot that I can do to stop it at that point. However, what if right up here we have another input, but this one, rather than delivering an EPSP, is delivering what we call an IPSP, or an inhibitory postsynaptic potential. What does that mean? It means that the ligand is opening up a chloride channel, a ligand-gated chloride channel. Chloride being the most important anion in this physiology of the neuron. And so we're letting negative charges come into the cell. So which one wins? Well, this is the idea of summation. Let's take all of my positives and let's add it up with all of my negatives, and we simply ask, who wins? It's completely possible that they cancel each other out. What might this look like if we're actually looking at the action potential on a graphic scale? Of course, we're sitting at resting membrane potential, minus 70 millivolts. And we've got threshold defined at the voltage, about minus 55, at which these voltage-gated sodium channels are going to open up. So if I hit threshold, I'm going to fire an action potential. So I'm at rest, and all of a sudden I receive an EPSP. So that's going to start to depolarize the cell. But before I get hit by all these IPSPs. What's going to happen? It's going to take me back negative. It might even hyperpolarize me. I'm down here. Well, maybe that EPSP fires again, and so I start to come back, right? It's a constant tug of war between all of the different signals coming in. Of course, maybe now I have a third signal. Let's say this one also delivers an EPSP. And maybe this is what I need to tip the scale, right? Because now I've added more positives, and as this comes in, I hit threshold. As soon as I hit threshold, those voltage-gated sodium channels fire, and boom, action potential. And that is going to propagate to the next one, and then to the next one, and then to the next one. And this travels down the length of the axon, ultimately ending up, just like we talked about before, in voltage-gated calcium channels opening, and then the exocytosis and release of neurotransmitter. So what we want to say now, right, as we think about all these EPSPs and IPSPs and the idea of summation, we've got two types. So we could have temporal summation, and we also could have spatial summation. Spatial summation is precisely what we just described. Spatial meaning everywhere, all of the inputs coming into this neuron. So the best idea of spatial summation is let's draw a line, and let's just look at all of the different regions where we've got signal coming in, right? We've got this signal up on top. We've got this signal coming in here. We've got this signal down here. And we're combining all of these coming in from different spaces, and we're determining what's going to happen. Temporal summation is a little bit different. Temporal summation is the same location, but its frequency. And we'll show you what that is in contrast to the spatial summation, which is simply the addition of multiple locations. So let's go back and look at what temporal summation might look like. So maybe this inhibitory neuron isn't firing, and so I don't receive that IPSP. Maybe I don't even receive this EPSP up on top. But I am receiving this EPSP down on the bottom. So it's getting me there. But you know what? It just doesn't provide enough. And so because we've got the sodium-potassium pump always in the background running, we've got this steady stream of potassium leaving the cell. It just wasn't enough to get us to threshold, and so we're starting to go back down. But then that same neuron decides to fire again. Like, maybe we'll get there that time. Not quite. But then it fires again, right? So this is the idea of the same location, just a different frequency. This same drum keeps on beating. And this time, that third one is enough to get me to threshold. And as soon as I hit threshold, action potential. So that's the idea of temporal summation. Spatial summation, again, being the addition of all of the inputs. Temporal summation being the steady beat of a single input. In reality, it's a combination of both of these things that eventually guide the neuron to know what to do, to fire or to not fire. And hopefully that gives you now a good idea of the physiology of a neuron and what it means to fire an action potential.